results of our drug screening campaign to identify pharmaceuticals that may be useful for treatment of GAMT deficiency. But before I start, I would really like to thank the organizers, and especially Heidi, to giving me the opportunity to record this presentation, because unfortunately I'm not able to attend this wonderful meeting. So, as I mentioned, I will speak about drug screening for GAMT deficiency by targeting AGAT, the AGAT enzyme, and specifically targeting AGAT expression. So what I'm going to do is I will briefly speak about GAMT deficiency, but very few words only. Then I will explain why AGAT is a drug target. Then I will tell you or show you the results of our drug screening campaign, and then I will come to some conclusions. So GAMT deficiency, as you know, is one of the creatine deficiency disorders, enzyme guanidinoacetate methyltransferase. The condition was first described by Sylvia Stöckler in 1994. It's a genetic disease, an autosomal recessively inherited disease, and many genetic variants, disease-causing variants, are known nowadays. One of the diagnostic hallmarks of the condition was brain creatine depletion, and you can see the MR spectroscopy that there was no creatine in the brain, and also phosphocreatine is significantly reduced. And all of that is because GAMT is the second enzyme in creatine synthesis. If this enzyme does not work properly, it leads to creatine depletion, but it also leads to the buildup of guanidinoacetate. That's the precursor. And guanidinoacetate has been shown to be proven to be neurotoxic. So when we ask what is the reason for the symptoms in GAMT deficiency, there are two aspects that go together. One is the brain creatine depletion, but on the other side, there is also, as important, the guanidinoacetate toxicity. So when we ask what is causing the disease symptoms in GAMT deficiency, then we have to consider two main contributors. One is brain creatine depletion, and the other is guanidinoacetate toxicity. That means when we look for treatments, we need to find treatments that do both, replenishing creatine and normalizing guanidinoacetate. So we proposed AGAT as a drug target because AGAT is the enzyme that regulates the rate limiting step in creatine synthesis. So inhibition of AGAT would lead to a decrease of guanidinoacetate and eventually also to creatine deficiency. But if one treats with creatine, then that would be fine. And we know for longer periods of time already that ornithine, for example, inhibits AGAT as an enzyme inhibitor. But we also know that creatine inhibits AGAT expression. And the third way we treat actually GAMT deficiency is by substrate reduction. That means a diet that is reduced in arginine. So all those three approaches are already used, but they are not efficient enough, and they are also very demanding, especially the diet and the poor taste of ornithine and so on. So that means targeting AGAT in a pharmacological way is the goal. There are two ways one can do that. One is by gene repression inhibitors. The other is by looking for drugs that inhibit the enzyme activity. In the presentation here, I report about our campaign about finding gene repressors. So our drug screening campaign consisted of 3 parts. One is the primary screen, where we had a cell selected the HeLa cell as a screening cell line, and we screened drugs from different libraries. All those drugs have a history of use in humans, and there were approximately 6,000 of those drugs. Secondary screen consists of testing dose response, IC50, and also looking for toxicity. And then final step is validating the drug candidates. And for that, we use 2 cell models. One is HAP1 and one is RH30. So first we had to select the right cell line that we can use for primary drug screening. And in order to find one, we looked for different immortalized cell lines and choose the one that had the highest AGAT expression. And this is HeLa, that is an cervical carcinoma cell line. And we had already experience in our lab with using this cell line for drug screening for a different inborn error of metabolism. So what we did is we bought a commercially available AGAT promoter and fused this promoter with a reporter, in this case, firefly luciferase gene. And this was then expressed in HeLa cells, so that if a drug is acting as a transcription inhibitor, it would reduce the firefly luciferase expression, and we can measure luminescence and use this as the readout for the drug screening. So we first tested several clones and then choose the clones 8 and 10 that express this construct stably. And then we tested whether our construct works. So we treated those cells first with creatine, because it is known that creatine reduces the AGAT expression, and one can nicely see that there is a dose response, while this construct does not respond to ornithine. As I said, ornithine is an enzyme inhibitor and it shouldn't respond. So that was fine. We used this cell line for the drug screening, and we used a total of 6 different libraries. They are mentioned here with more than 6,000 compounds. And that is the result of our drug screening. You can see we measure both the efficiency, so we hope that it reduces the luminescence, but we also want the cells still to be viable. That means those gray boxes here are the boxes where we're looking for candidates. So you can see there is only a small number, but at least we found 43 high score drug candidates that had a suppression of more than 3 standard deviations and were still viable better than -2 standard deviations. So these would be potential inhibitors. We also identified potential inducers, but we did not look into this more in detail. So then the secondary screen, we used the same cell line and we checked for dose response. And you can see there are a number of molecules that cause a nice dose response, while some others did not. So they would not really be useful for further testing. And then we wanted to see whether those 9 favorite drug candidates that we identified, whether they would have a synergistic effect to creatine. So what you can see here is those candidates tested with the same system, and as lower as the bar is, as bigger is the effect. So basically how much we can reduce the expression. And if there is a synergistic effect, that is the gray box, then it would be even less. So for example, if you look at acivicin, there is a synergistic effect. And then we used, this was with a concentration of 1 micromolar, and 10 millimolar creatine. And then we used the same with 4 micromolar. You can see there is a larger effect, but except for perhaps azivicine, there's almost none that has a synergistic effect. So not really much of a drug effect that we would hope, but there's another scale here, and that is this blue. This is basically toxicity. As darker, as more toxic the molecules are, and there you can see that, for example, azivicine, that has quite a nice effect here, is relatively toxic, and others like itavastatin even more toxic. So that was our secondary screen, but we also checked a couple of those molecules not in this HeLa promoter reporter construct, but in native HeLa cells. And there you can see, for example, itavastatin has a nice dose response with an IC50 5 micromolar and metrisone of 2.1 micromolar. And when we look at the effect of itavastatin compared to creatine, it has even a larger effect. But we also wanted to further test in after, so we also had to do validation. That was secondary screen. For that, we also had to use different cell systems and models, and I start talking about RH30, because a little bit later during our drug campaign, we did the expression study again to see which of the immortalized cell lines has very high AGAT expression. And there you can see HeLa, although we found this quite better than, for example, the HEK cells and the HG2 cells, is relatively low. And the largest expression was found in RH30 cells. So then it was very interesting to see when we investigated RH30 cells. So RH30 is a rhabdomyosarcoma cell line. When we investigate this further, it turned out that RH30 has very low creatine. That is what you see on the left-hand side. So these are the creatine concentrations. All of the cells in standard media cultured, and you can see RH30 has the least creatine concentration, but by far the largest guanidinoacetate concentration, while the other cells almost have no guanidinoacetate. So that was an interesting finding, and that is basically the quantitative representation of what I just showed you here, that RH30 has the lowest creatine and has by far almost 100 times higher creatine, guanidinoacetate than the other cell lines. So what does that mean? We identified an GAMT cell model, a cell model for GAMT deficiency. Of course, we would have liked to use patient cells for GAMT deficiency, but we don't have that. That is why we used RH30. So now we assessed, and the other cell model that we created is using HAP1 cells, but here we wanted to do something different than we did in the primary screen. The primary screen was just a construct of AGAT promoter and a reporter. We wanted to put a reporter into the endogenous AGAT gene. So we did this. We used CRISPR-Cas9 and inserted a nanologue reporter at the C-terminal of exon 9 of the GAMT, of the AGAT gene. So that is basically what we now have. It's similar, but it is the whole gene with the whole regulatory apparatus, and then it has this nanologue reporter, and then one can use this to see whether a drug changes the AGAT expression. And we have done this, we have published this, that this HAP1 AGAT NLUC insert works very well, because we can show that this AGAT cell line, this cell line responds very well to exogenous creatine by downregulation of AGAT, and it responds exactly in the opposite way to intracellular creatine. So it is a perfect model to see response to creatine-induced AGAT suppression. So that was what we used. And so when we did the drug validation with those cell lines, and this is the HUB1 AGAT NLUC cell line, what you can see is, unfortunately, so on top is toxicity, on the bottom is efficiency. And what you can see is that several of those candidates that we had are very toxic. While creatine is not toxic, and metrisone is not toxic, the others have cell toxicity. And that was after 24-hour incubation. When you look at the efficiency, that is what creatine does, 10 millimolar and 50 millimolar. So acivizine has almost no effect. Comfortizine has an effect, but we had to use much lower concentrations because of its toxicity. And so basically none of those had a significant treatment effect in this cell system. So all of the candidates were either non-effective or they were toxic. And then we used RH30. As I mentioned, we considered this as a GAMT model. So we wanted to see whether drugs can reduce guanidino acetate. That is, after 24, 48, and 72-hour treatment is creatine. And you can see, of course, if you treat with creatine, it goes nicely up. The other drugs have basically no effect on creatine, and it was not expected. But we had hoped that we would see much bigger effect on reducing guanidino acetate. And you can see there are only 3 drugs that have perhaps 30% reduction after 72 hours. One of them is creatine, that's what we know. The other is acivizine, and the last one is comfortizine. So, okay, they may have some effect, but not really very much. And we also checked whether those drugs have effect on AGAT expression. So basically all of this starts at 1, and you can see those 4 have an effect. So this is creatine, as we know, it is pitavastatin, it is etoposide, and it is comfortizine. So at the end, we identified perhaps 3 candidates. But when we look at the pharmacological spectrum, we can see comfortizine, for example, is an topoisomerase 1 inhibitor. So what it does is it causes DNA damage and apoptosis. One has to think about whether we want to treat our GAMT patients with such a drug. Acivizine has an impact on purine and pyrimidine biosynthesis. Again, could also be risky. Itavastatin or pitavastatin are statins, and statins are well known. They are HMG-CoA reductase inhibitors. They are used to lower cholesterol, but they do this by having an impact on cholesterol biosynthesis. So for a lifelong treatment, a GAMT patient would also be careful in doing this. So what are the conclusions? Based on our drug campaign that we did, and it took a couple of years, was very laborious. None of our drug candidates met criteria that would justify a further development as an investigational drug. So we are not advancing that. What we have learned is that the choice of an appropriate cell system for primary, secondary screens and for validation is extremely important. We would never again start with using a promoter-reporter construct. We would nowadays always start with a reporter that is integrated into the endogenous gene to find the drug candidates. But on the other hand, to say RH30, what we have chosen as a GAMT model, is perhaps not a good model for what we were testing, because one has to wonder, why is RH30 having such a high AGAT expression? Because obviously there is a dysregulation in AGAT expression, and that is why if we look for AGAT repressors, the system may not be the right one. So, the other thing, we had more candidates, and we always asked our pharmacologists about them. And turns out that almost all the drugs that had any effect on gene expression that we could identify had also significant off-target effects. So while AGAT is an excellent target to treat GAMT deficiency, to normalize guanidinoacetate, acetate, I came to the conclusion that gene repression as a tool to target AGAT is not a feasible option for the treatment of GAMT deficiency, and we need to find other ways to do that. At the end, I want to thank the people in my laboratory, especially Ilona, Mike, and Alex. Alex can also perhaps answer a couple of your questions that you may have. This was done in collaboration with Alessandro Dati from Mount Sinai Hospital Drug Screening Facility, and the pharmacologist who helped us with those questions was Shinaya Ito. I also want to thank the ACD for funding Alex. And so now I would say now the talk is open for questions, but unfortunately I cannot answer your questions. But thank you very much for your attention.
Alex, where are you? Oh, here, I see you. So he's there. So he can answer all of the questions that you have. Yeah. One here.
Hi, kind of same question as to another speaker. Was all the work done in male cells or male? Can you do it differently in? Does it have any sex implications?
Sorry. For this study, to my knowledge, anyways, all the work was done in cell lines. So as such, I don't think sex implications would play a role in the observation. It's unlikely, in my opinion, based on because these are cell lines, so they don't have gender.
So the question is whether sex plays a role in testing all of the molecules, and the response is that many of them are cell line, which is correct. The cell line, unfortunately, what they do, many of those that are used, have multiple chromosomes, some of which, some of them have multiple X chromosomes as well. So mean that to understand the role of sex in those cell lines is mission impossible because they screw up the chromosome. Okay. So that it is. So I mean, I don't. Okay. You had a question? No. Yes. Say again? Yeah. Some of them are male.
The HeLa line that he chose at the beginning is female, so there was at least one female line tested for sure.
Yeah, yeah.
Thank you. Okay, so when you did this work, I'm assuming that the library that you all chose was, do you know if that was like in-house or already approved in like Canada drugs, or are you also looking at other libraries to expand on, or from the hits, like looking at other isomers, or?
the project started before I joined the lab, and so the library selection, I don't have any knowledge about that. Sorry.
So the two drugs that he discussed were very well-known drugs, but and I agree. I mean, the problem is, I agree with Dr. Schulze, to be honest with you, that unfortunately, targeting gene expression is very hard. So you finish having side effect, basically. So what we are doing at I left the University of Utah, but we are still working on developing inhibitor of the enzyme, and that we hope it will be something that works. So we are progressing, but we are still not at the point of showing any data. I hope to have compounds that don't kill all the cell. Let me put it this way.